We're talking about the plasma membrane, and today we're going to talk about how things come and go from the cell. And a key term, whenever you talk about the cell, no matter what level you enter into talking about the cell at 6th grade or 7th grade life science, 10th grade biology, or an intro class in college, you always have to define the term selective permeability. Selective means choosy, right? Only certain things are chosen, and permeable means to pass through. So selective permeability just means that only certain things can pass through the membrane. So there's certain characteristics of molecules and chemicals and ions that can pass through, that allow it to pass through, because we talked last time about the structure of the plasma membrane and that it is made up of these phospholipids. And a lipid is a fat, and we know that fats and oils are impermeable to water, right? So water and oil don't mix. You have to shake up that salad dressing that contains the oil and the vinegar in it, right? So in order for things that are charged, like ions and sugars and salts and amino acids, to get through, they have to have some means of transport. They have to have a special transporter to get that through the membrane. Those are typically proteins that are embedded in the membrane that will allow those charged molecules through. So amphipathic is kind of a fancy word. You don't need to know that word. That's not something that is going to make you a better person after you leave this class. But it just means that it has two types of properties associated with it. It's partly water loving or water attracting, and it's partly water fearing or water repelling. So the word hydrophobic probably means water phobia, fearing, right? And hydrophilic, philia means love. So hydrophilic means water loving. So that's the attracting part. So when you look at the model of the plasma membrane, we say it's a fluid mosaic. And a mosaic, if you think of art, it's like the little tiles, and they're kind of carefully placed, and it gives you this really pretty kind of design using lots of different things or arrangements of those tiles. So this is like a mosaic. When I look at the surface of the plasma membrane, there's lots of different things embedded in it. It has a unique structure. And when we say it's fluid, it's because this membrane, because it's an oil, because it's made up of these phospholipids, there's lots of movement because of that. But luckily, we have a little bit of cholesterol embedded in among these phospholipid molecules that helps stiffen the membrane. So that's the function of cholesterol. It just helps stiffen that membrane so we can keep our cell membranes intact. Otherwise, they would break apart and be destroyed. So that's the benefit of the cholesterol. So other things we see in the plasma membrane are these proteins, and these proteins are going to help water-soluble molecules that come into the membrane but can't pass through. Some of them are transport proteins that allow substances to pass through. Some are enzymes that will break down substances on the outside and undergo chemical reactions that they don't even enter the cell. They interact with the cell, and then they move on and go and do their thing somewhere else in the body. So these proteins can be enzymes, they can be transporters. There's a number of different functions for them. We talked about last time about these carbohydrates. What are the functions of the carbohydrate chains extending on off the surface? Do you remember what we said? Yeah, cell recognition and communication. That's the function of those carbohydrate molecules, is recognition, communication. All right, so we have these proteins, and then the brick and mortar of the plasma membrane is what molecule again? The main structure? Phospholipids. Phospholipids are the most abundant lipid. It's the brick and mortar of the plasma membrane. It's what makes up the plasma membrane, right? See the phosphate head and the little two fatty acid tails? This is one phospholipid molecule. That makes up the brick and mortar of the plasma membrane. And again, what's the role of cholesterol? stiffens that membrane, gives it some stiffness so it's not so fluid. Okay, so then looking at the functions of these proteins, there's some that are just on the surface. We call those peripheral proteins. So anything that's just stuck on the surface would be peripheral. And if it's embedded and spans the whole length of the membrane, we call it an integral protein. Like this little one here that's just on one side, this one here, these two are peripheral proteins. They're on the periphery or the edge. That's just another word for peripheral is edge. And integral means they're integrated, buried in that whole membrane. So the different functions, like I said, are transport. Here we can see this molecule must be water soluble. It can't get through, so it has to bind to a special protein with the help of ATP, and it's actively transported across. So it's actively transported here because it requires ATP energy. This one is just a simple leak channel, so it's always open. It doesn't require energy. And this blue molecule just diffuses down from high to low concentration with no energy. And we're going to talk about those transport processes in more detail. Here's an integral protein acting as an enzyme. So molecules passing by the cell interact with that enzyme. A chemical reaction occurs, and this square is turned into a circle. Again, this is the beauty of biology and not chemistry, is we just use shapes for molecules rather than long, complex chemical structures. Here we just have signaling molecules. For example, a hormone can bind to a cell, a receptor, which is made of a protein, and tell that cell to do something. So here's just some type of binding some signal from the outside saying, okay, cell, do your thing.
Are these like molecules and stuff attracted to the cell based on what the cell says it needs? Or does it just like happen naturally?
It is a reaction with the protein, yes, but what brings it, what attracts it to the cell in general is this water-loving, these hydrophilic phospholipids. So that's what attracts it to the surface, and then it's attracted specifically to these proteins that match with the product that they're interacting with. Yeah, exactly. Like, you know how there's an active site on enzymes that are specific to that molecule? Like here we can see that active site for this specific molecule. Good question. Okay, cell recognition. We talked about some of these proteins will have carbohydrates extending off the surface, and that'll cause another cell nearby. So that's what this is. This is a cell with its own plasma membrane drawing near and interacting with this cell based on those protein and carbohydrate pattern on the surface. Here cells are joined together. Remember we talked about last time that there's these desmosomes. I said you didn't need to know that specifically, but there's cell-to-cell attachments. Remember we talked about the brain has tight junctions, so we can't let anything into the brain tissue. These are proteins that hold the cells together and keep them nice and tight. Like your intestinal cells, when you're digesting your food, you don't want spaces between your cells, so when the food comes through, it just leaks into your body tissues. You want those cells of your intestines tight. Have you heard of leaky gut syndrome that people have? That's when those cells are not tight together. There's some damage and these intestinal cells separate a little bit, and it causes inflammation and problems with gut health. And then they can just attach to the cytoskeleton and hold cells together. So if we're talking about cells in a tissue, like muscle tissue, the different cells are tethered together to their cytoskeleton, just holding those organelles in place and giving that cell integrity. So inside and outside, these proteins can attach. Okay, so that's proteins. So lots of different functions of proteins. Transport, enzymes, signaling, recognition, holding cells together, and holding an individual cell together. Then we talked about the carbohydrates are for recognition, cell-to-cell recognition, communication, so they can be attached to lipids or proteins, these carbohydrates. So if we look at our cell membrane again, some of these are attached to proteins, right? And some are attached directly to a lipid, so we call it a glycolipid. A carbohydrate attached to a lipid is glycolipid. And this is a glycoprotein because it's attached to a protein. Okay, so that's the carbohydrates. So here is an example of recognition. So when HIV attaches to a receptor on a T cell, that HIV can enter into the cell. But they found that some people lack this receptor. So what would that be an example of? Right here, right? Binding to a receptor and then allowing that substance in. So they find that some people, their cells lack this receptor. There's a piece missing, that little green CCR5 co-receptor on the T cells is missing, and the HIV cannot enter. So that means they've been exposed to HIV. It's in their blood, but it can't enter their cells. So therefore, it'll never destroy T cells, and they'll never develop AIDS. What's the bad side of that? Yes, they can still spread HIV, and that's where the HIV epidemic began. They have traced it back to one individual in the late 70s that had intercourse with lots of men, but that individual was resistant to the virus. So they didn't have the disease themselves, but they spread it to lots and lots of people. And we find that once people are HIV positive, it stays in their system, and we're going to study this semester. We're going to talk about exactly how viruses reproduce and how this whole AIDS things happen. But it takes about 10 years before the virus to multiply and kill enough T cells where people don't have an immune system anymore. T cells are really important for immune function, and once the HIV virus has destroyed enough T cells, then they catch everything under the sun. There's so many infectious things in our environment that we take for granted because our normally functioning immune system takes care of it, and we don't get sick from it. But if you don't have an immune system, you get everything. And the biggest one is a pneumonia and a skin cancer that is only for people with weakened immune systems. And that's some of the first symptoms of HIV turning to AIDS is that. For example, Magic Johnson developed AIDS in the 90s, in the mid-90s. He left basketball when he found he was HIV positive, and he had had unprotected sex with lots of women. That's how he acquired it, and he was very open about that. But because AIDS treatment has gotten so good, we are treating it like just a chronic health condition now, like diabetes. If you take your insulin and you watch your blood sugar, you're okay, right? If you're HIV positive now, if you take your antivirals, they can kill that HIV virus to where it's non-detectable in the blood. That means it's not killing T cells, and these people are not going to develop AIDS. So we've come a really long way here in the US with AIDS treatment, or HIV treatment. What? It is. What's the downside of that? Yeah, people are still spreading it because it's not a death sentence anymore. So if someone says, hey, I'm HIV positive, oh, well, that's okay. You know, you're okay, you look fine to me, it's not a big deal. So I'm a nurse in the jail, and we have people, inmates, that are HIV positive, and they're not taking their medicine. They take their medicine when they're in jail because they're stuck, right? And they take care of themselves when they're in jail, and we're giving them the medicine every day at the med cart, and they're taking care of it. But when they go back out and they go back on drugs, they're not taking care of they're not taking their drugs, and they're spreading the AIDS virus through those needles. So it's definitely in La Crosse. HIV is not something that's in the big cities, and you know, it's here, it's in La Crosse, people have it. So be very careful when you see needles. Like if you're walking downtown, you see needles on the side of the road, those are dangerous things. And you know, don't touch it, and just, if you see it, you can, there's like needle drop-off sites and things now. I'm not sure where they are, but we need to get better at that. We do have some locations for it, but not everybody uses it, obviously. If they're not taking care of themselves, they're not going to take care of the community, right? So be careful. Quick trip bathrooms too. There's needles people find in quick trip bathrooms. Can be dangerous places. Taco Bell bathrooms. Oh my gosh. Yeah, so you don't even, you know, touch those. Use a broom or something else to pick it up and put it in your needle drop box. Oh, yeah. Oh my gosh. Yeah. Put it in something like a empty water bottle, something that'll protect, because think of your person who does your trash, you know, they pick up that and it pokes their leg as they're, you know, rubbing it against their body. We have to really be careful. Oh, scary. Okay, so looking at then, this is selectively permeable, only allowing certain things in. So anything that is nonpolar, And nonpolar means uncharged. Okay, so it's not charged. We don't see that electronegativity. We don't see any charge on it. The electrons are shared equally. And these are uncharged molecules. Things like sugars and salts and ions are charged. They cannot cross the membrane without help. So you can add to this list sugars, salts, amino acids, ions. So sugars, salts, amino acids, and ions cannot cross the membrane. They need help. So what can cross the membrane then? Nonpolar things like, what are some examples? What good is it to say nonpolar if we have no examples, right? What do you think? What do all of our cells need every second of the day or we're dead? Well, we can go without water for a little bit. Oxygen, yeah, oxygen and carbon dioxide. Those can diffuse rapidly across. Those are uncharged gases. They diffuse right across, no carrier required. Something else that is uncharged is alcohol. Alcohol can freely pass through our membranes. Isn't that wonderful? Well, it's not good when you're trying to mask that you've been drinking and it diffuses across the cells of your lungs, out of your blood into your lung tissue and is exhaled, right? You can smell alcohol in people's breath. And when people have been drinking a lot and then their body breaks down that alcohol, they release another nonpolar substance called an aldehyde, and that you can smell on people's breath. And you know that breath of the hangover breath or the I've been drinking all night, now it's 6 a.m. and I got to go to work breath? Be careful with that, because I've been in class next to people like, whoa, you were drinking last night. You can smell that aldehyde. It comes out of their pores. It actually, you can smell it just sitting next to people. When I was in grad school, there was a guy that was just a big partier, a very, very bright guy, hardly studied. He had a drinking problem, actually. He got A's on every test. But you could smell that every single morning when he came to class. So, and I've had, when I've been working at the hospital, you know, we start at 6:30 in the morning on some shifts, and I'll have like a CNA walk by and just that breeze of, whoa, you know, they were drinking. And I only say CNA just because they're usually the younger people on the floor that are in that, you know, downtown crowd. So be careful if you end up, you know, drinking a little bit, you know, like around the holidays or whatever, if you are of drinking age, of course, be responsible, but know that that odor is on your breath, so you have to.
What's the, like, I guess it could be a longer question, but like, what's the process of actually getting it from? Is it that those alcohols are able to just?
Alcohol enhances a neurotransmitter that's called GABA, G-A-B-A, which depresses the central nervous system. It slows everything down, yeah. So the more alcohol you have, the more you enhance GABA, more the brain is slowed down and unable to interact with the environment, to the point you get to alcohol poisoning and the central nervous system completely shuts down and it stops breathing. And that's the danger when people drink too much. Okay, so these transport proteins then, if it's a charged substance like sugar or ions, it needs a transport protein. And some proteins are tiny little proteins just for water, and we call those aquaporins. So these are bolded terms that you should know definitely for our test. Aquaporins are tiny little transport proteins that allow water to pass through, because water is charged. Remember we drew a picture of a water molecule and it has that slightly selfish oxygen, which is electronegative because it pulls electrons a little closer to it. Remember we talked about cohesion and adhesion, those charged water molecules, so those can't pass through. They need a channel, so these are always open little channels called aquaporins. So when we talk about transport that is not requiring ATP energy, we call it passive. If something is passive, it means energy is not required. No ATP required. So diffusion is a passive transport process whereby things move from high concentration to low concentration. And you've experienced this before. If someone comes into class with a really strong body odor and they plop themselves down right in the middle of class, it doesn't take long while the person next to them notices that body odor. Then pretty soon Everybody in the back can smell that body odor, right? So that is just high concentration of those obnoxious particles, right? Those gas particles being released from that odor, from that bacteria, and it spreads to lesser concentration until the whole room has an equal kind of a smell of that body odor, right? Or if I put a drop of food coloring in a glass of water, it starts out as a dark blue drop, right? And then pretty soon the whole glass of water is an even color of blue. That's diffusion. Nothing is alive, right? There's no ATP energy being used. There's nothing living in that glass of water and that drop of food coloring. It's just random movement of particles. All particles are in motion. It's the law of conservation of energy. All particles are in random motion, just bouncing in the air, gases, bouncing off of each other, in liquids, you know, sliding past each other, but they're constantly moving and diffusing and spreading out molecules from high to low concentration. So there's no energy required for that process. So we say dynamic equilibrium. Make sure you underline or highlight that word. That's another important word I want you to know. Dynamic equilibrium just means that things are moving back and forth dynamically, which means not stationary. If I have a membrane that is leaky, things are going to move back and forth, particles, until it's equal on both sides. But it doesn't mean they just stop. It just means they're continually milling back and forth, but it's relatively equal on both sides. We don't see a mass movement one way or another. It's just a little bit of back and forth because they're in equilibrium. So here we put some food coloring in, molecules of dye. We have a semi-permeable membrane, but it's permeable to water. So the molecules are small enough to fit through this membrane. So the water molecules are going to move. We're going to see more of a net movement into where there's no molecules, right? So there's like what we call a concentration gradient. The gradient is higher on this side than this side. So we see this back and forth until it's equal. So you can see there's more arrows going to the right than there is to the left, right? So there we see a net diffusion to the right. And then when it's in equilibrium, a little back and forth, but it's pretty much balanced. So this is called dynamic equilibrium. It just means things are still moving, but it's equal. Yes? It depends on the concentration, yeah. And it depends on the heat of, there's a lot of things actually that determine diffusion. It depends on the concentration of one side versus the other, and like if there's more here compared to here. And then it also depends on the temperature, right? Things, what do you think would speed up diffusion, high or low temperature? High, yeah. So there's a lot of other factors too. The size of the molecules, all that. So this is just one particular molecule. Here's, if we add another solute in there, we see back and forth movement again, but more movement to the left of the purple molecule, right? And then by the end, when we reach equilibrium, both molecules are pretty much on either side in equilibrium, but there's still a little bit of movement back and forth. Okay. Oil and water, it wouldn't, right, we would see the water sit on top and the oil would be on the bottom because they don't intermix. Yeah, they don't dissolve because this is water, so it wouldn't dissolve. These are water-soluble substances. This might be sugar or salt, or like in this case it's saying a dye. But a solute is just anything that dissolves in water. So a solute you could just put after this sugar or salt or an amino acid. Those things dissolve in water. So substances diffuse down their concentration gradient. So the concentration gradient is just the difference in concentration between two sides. So this is the high side, this is the low side, so it goes from high to low. No energy involved with diffusion. So no work must be done. So we say no ATP required. So make sure you add that to that second bullet, no ATP required. No ATP energy. Highlight no energy. Okay, so we have diffusion of particles going back and forth. We call that diffusion. When we're talking about diffusion of water, that's called osmosis. So osmosis is the diffusion of water. Make sure you add a little highlight there. Diffusion of water across a selectively permeable membrane. So when water diffuses, it acts to dilute a concentrated space. So it's going to go from low solute concentration from a dilute space to a more concentrated space. So if we look at this example, where is there more solute? On the right or left of this U-shaped tube? On the right. So the water is going to diffuse through that membrane from where there's more water to there's less water. So would you agree there's more water on this side? It's more dilute, so it's going to go from the dilute side to the more concentrated side. And that's going to continue to happen until the solutes are balanced on both sides. But look we have more water on this side, don't we? Because in order to spread these out, we had to fill this with more water to get the concentration the same. So if we look at this through a microscope, here's the water molecules, right? The oxygen with the 2 hydrogens. You can see there's more water on the left side than on the right side. So when this comes across, when this diffuses, we have to pull more water molecules to the right side to get that concentration the same. So we call this process, when water is moving down its concentration gradient from high water to low water, we call that osmosis. So when you look at different concentrations of solutions then, we look at the solute concentration, where there's more stuff. So in this first example here, we look at this cell. Inside the cell, it's calling it a hypotonic solution, which means that particular environment where that cell has a low water concentration. I'm sorry, a high water concentration, low solute. So if something is hypotonic, if the solution is hypotonic, where we put a cell in, it means it's really it's not as concentrated in the solution as it is in the cell. So which way will water move when these 2 plant cells are put into a hypotonic solution? Yeah, water's going to move into the cell, and that's why those cells burst, or that water vacuole fills up nice and plump and you have a turgid cell. When a cell is turgid, it means it's full of water and it's plump and it's upright and it's a healthy plant. An isotonic solution means you have a cell that has an equal number of solutes compared to the environment outside of it. So in the middle, we see water going in, we see water going out. And that's because the concentration of the solutes in those middle cells are equal in the cell and the environment outside the cell. So iso means same. Remember isosceles triangle when you were in geometry or math class? 2 sides were the same, or the 2 angles. All right, hypertonic solution means you have a cell that is in a very concentrated solution. So what happens when you put a cell in a concentrated solution? Where does water go? Out. It always goes from high water, from the dilute to the more concentrated. So that's a hypertonic solution. And a real life application of this is what we do when we salt our steaks and let them sit there before we put them on the grill. Is it a good idea to salt steaks? What is that going to do? What solution are you creating outside the cells of that steak? When I throw a bunch of salt, you're creating a hypertonic solution outside that steak. It's going to pull water out of the cells and it's going to make your meat tough. So it's not a good idea to salt your steak when it's raw and permeable. So what you can do is sear the steak on the grill, seal those cells on the outside, then add your seasoning. Or some people rub their steaks on the outside with olive oil Then they season their steak, so they create an impermeable barrier for that seasoning to not pull all the water out, okay? And that's, yeah. Mm-hmm. Yeah, mushrooms are a perfect example. Mushrooms are a high water food. What do you call them? Vegetable? I don't know. Yeah, they're a high water food, so when you add salt to them, like you say right away, they shrink and shrivel. And sometimes we want that, right? Beef jerky is a classic example of shrinking and shriveling, and there's a lot of salt, right, in beef jerky, and that's what pulls all the bacteria out too, and that's why it's a great meat for people to keep long term. You know, in the old days, you know, they would salt and smoke their meat, and it would stay for months, and that's how they survived the winters and things, you know, by eating buffalo and preparing all that meat and turning it into a jerky. So when a plant cell has lost so much water because you left the plant in the corner of the room and you forgot about it and you forgot about it, and all of a sudden you look and say, oops, it's all shriveled and crunchy, you can't bring it back. When plasmolysis occurs, it means that the cell membrane pulls away from the cell wall. Can you see how this has kind of pulled in here? So here's the cell wall, and this is the plasma membrane. When it pulls away, the cell dies because it can't transport nutrients anymore with the plasma membrane pulled away from the cell wall. And then it's time to throw that plant in the garbage or throw it out in the yard and recycle it. So we have, now you tell me what's going on here. So we have a cell, this little balloon-looking structure, that is 0.03 molar of sucrose and 0.02 molar glucose. Don't get thrown off by the M and the 0.03. It's just look at the numbers, which is bigger. Look at that as money. That's 3 cents versus 2 cents, which is bigger? 3, right? And then outside is 0.01, so 1 cent. So the outside, is it more concentrated or less concentrated? The outside, is the outside more concentrated or less concentrated? It's less concentrated, right? 0.01. And even if I add all three of those together, it's still 0.03, and if I add the two together inside the cell, it's 0.05. So it's definitely less concentrated outside the cell. So the environment is what type of solution then of your three choices? Hypotonic. The environment is hypotonic. What is the cell? Hypertonic, right? Hypertonic. So which way, if I told you that the cell is permeable to glucose, which way would glucose move? Into the environment. It would move out of the cell, right? If I told you that the cell was permeable to water, which way would, and maybe draw an arrow so you know what direction things are going to move. So glucose is going to move out of the cell. If I told you this cell was permeable to water, which way would water move? Into the cell, because remember, it moves from the dilute environment into the concentrated environment, because it's osmosis is the diffusion of water from higher water to lesser water. So water is going to move this way into the cell. So draw a little arrow and draw your water going into the cell. So if sucrose is impermeable, that's going to draw more water in, isn't it? Because sucrose can't move out. So that's going to draw more water in to dilute that sucrose until what? How much water would flow in to dilute the sucrose? What would stop the flow of water in to dilute the sucrose? Dynamic equilibrium. When would equilibrium be reached? At what level of sucrose inside the cell? So you're adding more water in to dilute that, right? Just like if you make yourself Kool-Aid and you taste it, oh, that's too strong. Or lemonade and you add more water. Water's going to keep flowing into the cell until what level of sucrose is reached? 0.01, until it's the same as the environment, right? Dynamic equilibrium. So this is going to continue to flow in until this number drops to 0.01, and we have a more dilute sucrose. So what's going to happen to the size of my cell? It's going to get bigger, right? It's going to get bigger. And that's what happens in our own bodies. You know when you're dehydrated, right? How do you feel? What does your mouth feel like? Dry and sticky, right? And if kids are really dehydrated, or let's say your dog's been vomiting and diarrhea, got into something in the backyard, or my dog's got giardia this summer. And I raise a dog breed called the Brussels Griffon. I'll show you a picture sometime. I talked a little bit about it in lab, I think. Anyway, we have goats, and we had a lot of rain this summer, and somehow everybody got sick, and it was just diarrhea central at our house. It was really gross. So I was giving a lot of electrolytes, a lot of medicines. We finally got through it. But how do you know if you get a dog that has vomiting and diarrhea, how do you know if it's time to take them to the vet? You look for dehydration. And one thing you do is you pinch their skin up. You just pinch the skin on their back, in the middle of their back, pinch it up. And if it sticks together, that's called tenting. And it's because there's no fluid under the skin that they're dehydrated. Then you would make sure you take them in to be seen because they need IV fluids at that point. And that's what happened to our silly goat. I had to have the vet come and give it IV fluids to keep her alive. And then she survived, and everybody's healthy now. But it was a real hassle. So when cells take on water, they're plump. When they lose water, obviously they shrink, and we can feel those side effects in our own bodies. So when you don't hydrate enough, your brain cells actually shrink a little, and they're less functional. So if you come to class, you haven't eaten breakfast, all you've had is coffee, and you haven't had any water, and you've got a big test to take, you're not firing on all engines because of shrunken brain cells, because everybody dehydrates a little bit overnight. Have you noticed that? If you get up and walk out the door and don't drink anything, you're like, oh my gosh, I'm thirsty this morning, right? So always bring a bottle of water to class. Make it a habit for just healthy living is to get yourself a nice water bottle you can be proud of, right? They're not that expensive. And even if you buy one at Quick Trip for $2 and keep refilling it, that's fine. But plump up those brain cells, and you'll find that you're functioning better when your cells are hydrated. So a term when a plant is plump and has a nice full central vacuole, we call it turgid. And when it's not, the cell becomes flaccid or limp or wilts. And when death occurs, that's when plasmolysis sets in. And this is a plasmolyzed cell where the cell membrane pulls away from the cell wall, and that cell's a goner. And we had a beautiful plant my husband got when he was in the hospital for his neck surgery years ago. And I was really taking care of it, and I was really proud because I'm really bad. I do not have a green thumb. And I made the mistake of putting it around the corner of a room that we don't spend a lot of time in and put it in the window. And it just fried there because I forgot about it, and I was so disappointed. And I thought, oh, I'll save it. I watered it, and it did nothing. It just was crusty and gone. So plasmolysis definitely set in. So this would be a flaccid plant that maybe isn't getting enough water. We have equal back and forth. We need to keep watering a plant. It needs to be in moist soil, so it's constantly taking in water. Because where do plants lose their water? What structures in the plant are constantly giving off moisture? What do you think? What parts of a plant? Just think big picture now. We're mincing hairs right now, but let's zoom out and just common sense street knowledge. Yeah, the leaves. So the leaves are constantly giving off water. So the roots should constantly be taking in water. So we should always see a hypotonic environment in that soil that's going to continue to pull water into the plant. We don't want an exchange going on. We want constant take-up of water because they're constantly losing water off the leaves. Okay, the next topic then is facilitated diffusion. Facilitated diffusion is still movement from high to low concentration, but we need a channel protein. These things cannot take in anything charged because that membrane, we're talking about a semi-permeable fatty membrane, can't take in things, so there's aquaporins and ion channels that have gates that are not always open. It only opens when the cell needs those ions to perform a function. So here's an example of facilitated diffusion. We have more solutes on the outside than the inside, so it just flows right through that open door, no problem. Here it has to open up and carry it across, so it's not just an open leak channel, just a doorway. It has to actually interact with this carrier protein, change the shape of the protein, and then it dumps it on the other side. But are we still going high to low in both of these examples? Do you see higher on one side, lesser on the other? Yeah. So we call it facilitated diffusion, meaning it's passive. Diffusion is always passive, but facilitated means it needs a little help. You can't just, it's not simple diffusion where it can just diffuse, like oxygen, carbon dioxide, alcohol, nicotine is another one that can diffuse right across. These things need a little help, so they need a protein carrier.
So the bottom one is
They're both facilitated diffusion. They need a protein. Both of these examples involve a protein. They both are going from high to low, so there's no energy required. So this is just an open door, and this is a carrier protein. So two different proteins, channel protein, carrier protein. Yeah, they are different types of proteins, but the fact that it's high to low means there's no energy needed. It just happens naturally with chemical reactions, not with physical ATP energy. Now, active transport is when we're going from low to high. Just like when you go up a slide, to get to the top of the slide from low height to the higher height requires energy to walk up. And then you get to the top and you just slide down, right? High to low, no energy required. But when we're going opposite that, we do require energy. So where that energy comes from is this special molecule called ATP. We're going to talk about that process in a lot of detail in the next unit. But for now, just know it's an energy molecule used by cells. And it's going against the concentration gradient. So when we say against, we mean it's going from high to low. I don't know if it'll let me. Let's see. I think I have to like, what? I did. I'm terrible at printing low to high. Okay, so ATP is the key thing here. We need energy, requires energy when we're going against concentration gradients. But we have proteins to take care of business. So here's an example. Here we have sodium that is in low concentration inside the cell. It's in higher concentration outside, because we can see here it's telling us the sodium concentration outside in the extracellular fluid is high. So it needs a special pump. ATP has to bind to that pump in order to transport it out. So we take a phosphate off of ATP, we plop it on this protein that energizes the protein and dumps that from low to high concentration. Same thing with potassium. Potassium is in higher concentration inside, lesser concentration outside, and it dumps it into the cell against its concentration gradient. So a perfect example of active transport is the sodium-potassium pump. That's a classic example of active transport, taking sodium and potassium and moving them from low to high concentration. We're going to talk about that in the next unit. Yes, you have to kind of take it at face value right now, but you'll understand it a little more in the next unit. Good question, though. So passive transport, again, in summary, is high concentration to low concentration, no energy required, right? No ATP required for either of these. So whether it's diffusion, simple diffusion, or passive diffusion, simple diffusion or facilitated diffusion, no ATP is needed. But in active transport, we need that ATP molecule. Now, other types of diffusion that we're not going to spend a lot of time on is co-transport. Sometimes we have ions, solutes that are transported. Two things are transported at the same time across the membrane, co-transport. So here we see hydrogen and sucrose are being transported together with the help of two different proteins. You don't need to know the details of this, just know that co-transport is another type of active transport. So it requires ATP. So ATP is needed. This is horrible handwriting, sorry. But this allows me to keep it on the computer for the video. So we need ATP for co-transport. So that's what this is. Bulk transport is when we are looking at exocytosis and endocytosis. So exo means you're taking things out of the cell, and endo means you're bringing things into the cell. So whenever a cell is taking in, like that example we showed earlier, taking in bacteria, an immune cell is taking in bacteria, that's endocytosis. So here we can see things. This one is a cell that is making maybe a lysosome, and now we're going to dump that out, or maybe it's making an enzyme, it's going to dump it out. This is exocytosis. So it's a membrane-bound substance inside. This is a vesicle, right? And then that fuses with the plasma membrane, and that substance leaves the cell. Exocytosis. Here's another example. Here's the rough ER, the Golgi apparatus, taking that protein and dumping it out of the cell. Exocytosis, it's leaving the cell. Endocytosis is bringing it into the cell. So it's the opposite of exocytosis. And there's three types of endocytosis. There's phagocytosis, pinocytosis, and receptor-mediated. Phagocytosis means cell eating. Pino means cell drinking. Think of Pinot Noir. It's a type of wine you drink. So pinocytosis, cell drinking, so it means it's taking in liquids. And phagocytosis means the cell is taking in solids. So if it's a solid, like bacteria, we call that phagocytosis. And if it's some liquid substance, then it's pinocytosis. And receptor-mediated just means that substance has to bind to a receptor first. So here's phagocytosis. There's a food particle. Here's a liquid particle for pinocytosis. Again, these are coming into the cell. Right? These are all examples of coming in. And here it has to bind to a receptor. So the requirement here is a receptor. In order for that to bind. And then again, it can come into the cell. All right, so that concludes